Okay, right. So thank you, David, for inviting me to come and give this talk. And as the last kind of proper talk at the conference, it's quite a hard one, this one. And I actually I spent quite a few hours during the conference writing these slides, because it's not a talk you can write in advance, because it has to reflect on what everybody said. So I'm thinking about the future. I'm thinking about what the research challenges are that we need to address within this community. I guess to start with a little bit of a background reminder, I mean, I think I don't really need to pick this up there. These numbers are way out of date for COVID-19, but, you know, it caused huge numbers of cases and deaths around the world. I suspect the death toll somewhere in the like 20 million space now. They know that the numbers that are estimated on there are way under what it should be. But it's not just the only thing. If we look at WHO leading causes of death, we find lower respiratory infections in there at number 4. And we find lots of others of these which are affected by air quality. I think TB might be now number 11, but it used to be in the top 10. So there's respiratory infections worldwide, and air quality is part of that bigger picture, and the pandemic really mean that this is a huge health challenge worldwide. And then it's not just those respiratory infections. When we start thinking about how does disease transmit in the air, yes, there are those communicable diseases that we know are airborne. So TB for a long time, measles, COVID-19, influenza, etc. But when you start looking at literature on hospitals, you find other pathogens in there: Pseudomonas, Aspergillus, non-tuberculosis mycobacteria, pneumocystis, all sorts of others in there, which there is evidence for transmission through the air. And then we also find a weird airborne role in other things as well. So things like MRSA, which is associated with wounds, yet there's evidence for aerial dispersion in environments, contamination of surfaces. Norovirus, there's loads of data out there which shows that norovirus gets aerosolized and it will survive for very long periods of time in the environment. So we're not just talking here about human respiratory sources, we're actually talking about sources from water and waste. For some immune-compromised patients, we're talking about outdoor air, we're talking about resuspension off surfaces. And of course, we know that Far-UV can interact on surfaces as well as in the air. But we have to remember, going right back to the beginning of this conference and the talk that Shelley gave, we have to think of that much bigger picture. So GUV is one of those factors that we can put into buildings, but buildings are designed with a whole raft of different aspects and a whole push and pull of challenges, some of which are there because of regulatory things, so net zero, climate agendas, airtight buildings, etc. Energy is always going to be there. It's the reason for that energy efficiency will change over time, but that's always part of it. But then things that are much more nuanced around human behaviours. So things like noise, security of a building. Do you open the windows or is it secure to do so? What about air pollution inside and outside? And comfort. I don't know if anybody in here, there's a few people who've got CO2 monitors in here. The air quality from a CO2 perspective is really good in here. Okay, the temperature looks really good. The humidity looks really good. Is anybody a bit chilly? Yep. So it's going to have velocities in here which are a little bit too high and you can feel them. So we've got a set of metrics, but they don't always tell us the right things. So that's the bigger picture space. So one of the things in the UK that happened as a result of the pandemic was a piece of work that was commissioned by our government chief scientific advisor that I was quite heavily involved in. And this was led by the Royal Academy of Engineering and called Infection Resilient Environments. It was a 2-phase piece of work. The first phase was very much emergency. What do we need to do to buildings during the pandemic? What messages do we need to give out to people? The second one was, where do we look to the future? And we did quite a substantial piece of work around this. We looked with focus groups and sort of expert advice. There was a literature review piece, research review piece, and a cost-benefit analysis. And the word cloud there, which you can see a little bit of, was some of the issues that our the focus groups raised around what does the poor infection resilience in buildings give us. Now, bear in mind this was done during the pandemic, so maybe they had a slightly different focus, but there were all sorts of things in there, not just about disease, but things around how it might change things. For example, you see increased car use. That was public transport, people changing their transport modes and things. And we released a couple of documents on this. I can give you the link later. It's a really quite a good read. But then one of the things that was done as part of it was commissioned a piece of work around the cost benefit of ventilation. And this looked at the risks associated with infection, transmission, and that was in terms of illness and absenteeism from workplaces and things, versus the cost of putting improved ventilation. And this focused on ventilation rather than their cleaning technologies, but essentially showed that there was a benefit to this, particularly in community buildings, so schools, hospitals, etc., and in many commercial buildings, less so in residential. And I think that plays out with many of the things that people have talked about here. But this tallies with work that's been done over 20, 30 years ago in the US, which says if you improve indoor air, you'll improve health, you'll improve productivity. There's a big cost benefit for doing it. So, okay, so that takes us, that's that big picture piece. That now takes us into where do we go for Far-UV? And I'm going to go right back to what David said in his talk at the beginning, because ultimately, does it work? Is it safe? Those are the big themes of the entire conference. And that's what we're all really kind of working towards. And I think, David, you said at the beginning, does it work is almost the first one, because if it doesn't work, the second one is a bit pointless. But we have to know both. And so I'm going to ask the question, well, what do we still need to know? Okay. And I'm not going to cover everything that we still need to know, because I don't think you could sit there for that long. But I thought, well, how do we understand Far-UV? And maybe I'm coming to this from my perspective a bit on the work that I've been involved in. But we carry out a range of different studies. Some of those are very fundamental, small-scale laboratory studies. How does our microorganisms behave? Or what happens in a very small chamber from a chemistry perspective? What happens in terms of skin and eye studies, in terms of, again, very controlled settings. And then gradually, we sort of scale it up. Can we go to controlled chamber studies? We go to modeling studies, and then the real world. And these get more and more complex and messy as we go through them. So I'm just going to touch on a few points from one or two of these areas. And the first one I'm going to mention about is inactivation. So a lot of the focus over the past 3 years has been there because of COVID. We've been looking for, most of us cannot work with SARS-CoV-2, so we've looked for surrogate pathogens. And quite a lot of studies have looked at these. The vast majority of inactivation data is from aqueous solutions or on surfaces. And this is just two examples for comparing 254 on the left and 222 on the right, all showing relatively similar picture, although on the whole, it's about, you need about half the dose to get the same sort of activation for 222 than you do for 254. When we think about air, I think David might recognise this one, and Manuela. There's a few pictures in here I've been borrowing from people's papers. But we use these very small flow-through chambers and to measure microorganisms generated with a nebulizer, usually a Collison nebulizer, aerosolized, passing through a flow chamber to measure inactivation. And there's actually relatively few microorganisms that have been characterized in air for 222. Those which have been a bit more focused around the sort of pandemic space at the moment. We heard earlier in the conference a question about fungi. I haven't seen any fungal data. Maybe there is some out there, but I haven't seen yet, but I haven't seen any. We haven't really looked at things like temperature and humidity. And if we go back to 254 nanometer, actually there's lots of that characterization happen. But even within that characterization, those flow things are complex. And as I was putting this together, it got me thinking, and it took me right back to nearly 10 years ago when I first started working on UV. And this was the very first project I ever did on UV, where I was doing computational fluid dynamics models of the flow-through chamber that we created in Leeds to do decay constants for various different microorganisms. And in that, we looked at what actually happens in this chamber. What does the flow look like? It was quite nicely laminar in this one, but to start with, it wasn't. We had these weird recirculations in there. But you've got the top right is that was a surface measured irradiance field that was then put into the model. You can see it's not uniform. You've got this non-uniform distribution. So just assuming a uniform fluence rate might not be the case. And then actually in the model, you can see on the bottom right, that's the concentration of microorganisms passing through that. And you can see you get a non-uniform disinfection rate or inactivation rate as they pass through. So actually the design of the chamber and the flow of the chamber and the UV light intensity distribution will determine what inactivation constant you calculate. And that makes it quite a challenge. So I think this is one of the areas that we need to think about, because we need that good data for anything we model. Yet actually, how do we compare between different test rigs and different labs? How do we characterize all of those different parameters? There's a huge number of parameters you have to characterize in one of these test rigs to get it right. Which microorganisms have we done? Which should we be doing to make sure we've got that breadth of information that we need? And then what do we suspend it in? Do we suspend our microorganism in distilled water, in ringers, in bovine serum, in artificial saliva? Do we do it with a Collison nebulizer that typically generates 2-micron size aerosols, or do we generally use something else that is a bit more representative of a human source? What temperature and humidity? Do we need to do some variation, different ones in there? And then even within that, we get different inactivations. What is the fundamental mechanisms that are explaining those? So I think there's a whole set of actual questions, all based around really quite what appears to be a simple benchtop experiment, and actually it's a really complex benchtop experiment. And that's again why we need to bring all these different places together. So we've done quite a bit on chambers, room scale chambers. We can go to room scale. Room scale is great because it's much closer to the real world, but you've got that control over conditions. You can start looking at different lamp placements. You can look at room layouts. You can mock up a hospital room. You can put different microorganisms in there. You can put some chemical species in there too, etc. And these are just a couple of pictures of some of the things that we do in our chamber in Leeds. How do you do experiments in the chamber? So well, there are two different ways of doing these. Essentially, you are nebulizing a microorganism in, same problem we've got with the Collison nebulizer I had before, but you're trying to determine what happens in there. So we do two different approaches. We usually use what we call a steady state test. So we'll nebulize that microorganism into our chamber. Usually our chamber is ventilated, so we'll have the mechanical ventilation running. And we'll take samples when it's under steady state. And what we see is even under steady state conditions, we get variation. We get variation because actually the real world mechanical ventilation isn't steady. It's got fluctuations in it. And it's biological, and as anybody's ever done a biological measurement in here knows, if you measure something, you'll get a different answer every time within a boundary. So we get these fluctuations, and then we can switch our UV lights on, or whatever, leave it for a little bit, take another set of samples, and we've got two conditions, with and without. But crucially, we keep nebulizing all the way through the experiment, regardless. And that means that it's representing a room that's continuously occupied with and without a UV. The second one is what's often used, probably because of some of the EPA regulations about demonstrating how we get to 99.9 whatever percent inactivation, and it's a decay test. And essentially you can do a decay in a room. You have to do a control without a device present to get the natural decay. And then you can do it with a device present, and you should, hopefully, if your device does its stuff, you'll get a more rapid decay in there. So you can take samples during that decay. You have to repeat it, because if you just do one experiment, you've only got one set of data, and you have to get replicates in there to get reliable things. And you can get a difference in the decay rate, which will give you that efficacy of the device. And it'll also tell you the time it takes to reach whatever inactivation state you want to get to. We normally use steady state. And the reason we do that, particularly for GUV, is that inactivation, that decay, is so fast that we can't take enough samples quickly enough and accurately enough during the decay period to actually measure it with any degree of accuracy. So that's why we go down the steady state route. But there's a number of studies out there. Different ones have done different things. To my knowledge, for 222 nanometer, there are only 2 chamber studies. So one is from the group in Hong Kong, City University in Hong Kong, who have used a chamber that's about 12 cubic meters, I think. They actually set it up with their 222 nanometer device to do an upper room. So they're only irradiating the upper room rather than the lower zone. And they carried out decay tests with some different bacteria. And you can see the decay rates there. And they use that to calculate inactivation rates. Whereas our experiments say we use this steady state methodology. We are irradiating the whole room rather than just the upper room with 1 or 5 lamps. And you saw earlier in the week, our researcher Wasim, who unfortunately couldn't travel to be here, was able to measure under different ventilation rates, different numbers of devices, and to be able to show this inactivation with this continuous contamination. And from that, we calculated, this was with Staph aureus, which bacteria, an inactivation constant of about 30. And this is where we get a conundrum, because I had to put the graph on, because everybody else has put this graph on. So I almost was going to put a QR code for you to give us royalties on the graph, but you know, this is where we actually end up with a conundrum, which has been alluded to in several of the talks. We see a higher inactivation than we'd expect to see. We basically calculate an inactivation constant that's about 10 times higher than we think it should be. And you can't, explaining it is really hard. Now, the 2 studies that do that are our chamber, which is ventilated, it's got a mechanical ventilation system, and the air that it's ventilated, it's HEPA filtered, but it is just outside air. And the other study is the one that Manuela presented and David alluded to, which is in the murine virus, which again is a real environment with real air. Everything else are these tightly controlled chambers. So what explains the differences? I think that there's an open question there. If anybody knows the answer, please tell us. Back to Kenny's plea. But I think there are some open questions about why do we see differences in different size chambers? Why do we see these performance differences and things? And then when we think about chamber experiments, there are again a whole set of challenges in here. Chamber experiments that don't come easy. They are specialist facilities. They're biological facilities. We have to do these experiments with the chamber sealed, and all of your samples have to be taken from it externally in it. You need expertise who knows how to do that. They're time consuming, etc. And sometimes they won't work. You will do a set of tests and you'll get nothing. But we have very little data so far at chamber scale for 222 nanometer. And so we don't know, there's a lot we don't know around room layouts, temperature, humidity, the makeup air into the room. And then again, as I said before, the suspension fluids, how you nebulize, how you create an aerosol. Can you intervene when people are closer together? We've done a little bit of initial study on that one, which we haven't got the graphs in it. Yes, it does still work, but not as effectively. But I'm a bit reluctant to say too much on it because we need more data. I think some microorganisms are really hard to work with. So some things, one of the reasons we use bacteria, it's very easy. Some of the viruses are really hard to work with, really hard to get to a high enough concentration in such a big space to measure inactivation. And then, of course, we can couple inactivation and chemistry, although I don't know that we can do them at the same time, because when you do an inactivation experiment, you deliberately put something in there that will change all the chemistry for you. So there's a whole set of complexities. I touch on modeling. We've seen several people talk about modeling here, usually computational fluid dynamics, sometimes zonal airflow models. We can, within those, they have some real power because you can put those 3-dimensional UV fields in there through different methods, but they will all give you something fairly similar. You can model tracks of particles, you can calculate the cumulative dose, you can calculate inactivation rates, you can explore factors around design, where people are located in a space and things. And I've just shamelessly stolen a couple of pictures from Kenny's presentations earlier to illustrate this. But they're challenging. So models are all an idealized version of the real world, and getting the right boundary conditions on your model is hard. They nearly always involve something very static, whereas actually people move about in the real world, and buildings are much more variable and complex. People's emissions are complex. We have changing conditions. So the picture you can see on the right is a multi-zone hospital ward. It's using Contam to model a tracer through there to represent an infection. And the variability you can see over several months is solely due to the weather conditions in there. Nothing else. That's the only variability. So you can see, well, it can vary by 2 or 3 times depending on the weather. And that's very hard to capture into models. Putting in both inactivation and chemistry together, I think there's some real possibilities in there. But again, it requires that expertise to do it. And then, of course, with any model, how do you validate that model and know that it's right? So really teaming up with people who are doing small-scale bench experiments, chamber studies, etc. And I also want to talk about real people breathing out, because the exhalations that we produce, bear in mind we're thinking about respiratory diseases here, everyone's different. We produce this spectrum of particle sizes. We don't just produce 2 micron particles. They range from under a micron to over 100 microns. That size distribution depends on you, depends on how you exhale, depends on the angle of your head, depends on whether you cough or speak, depends on the temperature and humidity of the air, and of course these evaporate. And what here is showing some CFD simulations of an evaporating model. So you can see the particles, if you look at on the left, you get some big ones which deposit out, and then this jet which comes out and goes with the airflow as a buoyant plume and then disperses around the room. And on the right, these are little tiny diagrams, but the sort of spike you can see is a person standing and they're coughing. And on the top, these are the really big droplets that just behave ballistically, come out of your mouth and land. On the bottom are the really small ones, so they evaporate, become really small, and they disperse around the room. And then in the middle, you've got this bunch of intermediary particles, some of which deposit, some of which stay in the air. We're not capturing any of that at the moment in any of the models that we're doing in the GUV space. So now I'm going to touch on the real world, and this gets even more complicated, but we need a whole bunch of different things in the real world. We need to think about the bioburden and the chemistry, as we've heard from several talks. We've got the whole acceptability piece, which Kate's just talked about, and the messaging that comes into that. We need to think about practicalities. How robust is this? Can we maintain them? How usable is it? We need that long-term data on people's exposure. And then Don talked about this the other day around those challenge studies. So we need to actually measure some infection endpoints in here, because we can't just, I think we all know from the bioburden work that it works, but we do need some of those long-term infection endpoints as well. And Don alluded to some of these the other days, the Wells studies, TB studies, and most recently the ones around TB in Peru, and there's another one in South Africa, which looked at those measurements of, these were human, the middle one and the one on the right, human to guinea pig studies. The one on the left is the measles study, which is infection rates in classrooms. We're actually carrying out a study at the moment in primary schools in the UK. This was a pandemic study. This was looking at air cleaner interventions, so HEPA filters and 254 nanometer UV. It's been a really complex study. We've got 10 classrooms which are a control group, 10 which receive air filters, sorry, 10 schools which are control group, 10 which receive air filters, 10 which receive UVC. We intended to do upper room UVC and immediately discovered at least half the schools had too low a ceiling, so we had to put active air UVC into some of those, and we hit regulatory problems. We are measuring indoor air quality parameters in every classroom, because we need to say whether the ventilation is different in different classrooms. That means we've got something like 540 classrooms which have got air quality monitors in them, and we measure illness absence every week there. And it's been a long study. We're just in the process of publishing at the moment. I can't tell you the results, but they're looking good. it's, there were complexities of running these and doing this in the real world and putting them into schools where the schools have the control and we don't, has been a really interesting and challenging experiment. But these experiments and studies need to happen. So I'm kind of going to finish up by talking about some of the risks and benefits. And we've heard quite a bit of discussion over the past few days around this risk-benefit aspect. So we know there's a huge potential to reduce exposure to pathogens, reduce infection risk. And if you break that transmission, you don't just change that for the individual, potentially there's a knock-on effect onto the population as well, because there are less sources and therefore you reduce, as somebody said at one point, you flatten the curve. But we know the secondary chemistry could be a risk. We don't think there's a microbiome risk, but I think there's still a question mark in there a little bit. And how do we trade this off for different settings? So just to think this through a little bit, what about healthcare settings? If you're a patient, you're there usually for quite a short period of time, maybe a few days, but you are in that room, in that hospital for 24 hours a day. Your risk of hospital-acquired infection is quite high. And in the box on the right, I've just highlighted some of the UK data. So we had over 290,000 people in UK hospitals in over a year who had COVID. And the estimate from somebody who gave this to me the other day was that about 28% of those caught it while they were in hospital. So your risk of catching COVID in a hospital is really quite high. And that's without thinking about all of the other infection risks in a hospital. Most hospitals, even in the UK, which are poorly ventilated, have some background ventilation that I would say is reasonable. And so because of that short duration, maybe your exposure to the air chemistry is quite a short period of time. So perhaps the infection benefits dominate at this point. I haven't calculated it, I'm not sure, but I would hazard a guess that it might. But what about if you're staff? So you're not exposed 24/7, you're exposed for maybe 6-8 hours a day, depending on how things are set out in a hospital. If you've got a poorly ventilated space, your exposure to the air chemistry could be more significant. And of course, this might go on over many years, but your infection risk's also high. And if you look at the graph, this is relative rates of COVID-19, healthcare professionals are way higher than anybody else. So, if you're a healthcare professional and you're going to catch COVID maybe once every year, maybe every 2 years, for the next 30 years of your working life, where's the trade-off at that point? And I don't know. I think that one is a more complex trade-off, but it's not an easy one to bottom out. So we need to be able to do this properly. And I know Jose shared some initial discussion around this and got us talking about this. I think we need to think this through in a more complex way. We can use quantitative microbial risk assessment or quantitative chemical risk assessment approaches. There's some well-established approaches out there. But thinking about different population groups, different settings, what is that exposures? And then perhaps thinking not just about mortality, but morbidity effects as well in there. Because, for example, COVID-19, and of course this isn't the only infection we should be thinking about, we've got this challenge of long COVID on top of the acute infection itself. And of course, what about the next pandemic? We don't know what SARS-CoV-3 looks like yet. Let's hope it's not any worse. Back into that bigger picture, we need to know more about how this fits with everything. What's the sustainability of it, the impacts on materials, and I don't think anybody's mentioned climate change, but how we use buildings is going to shift. Our air quality is going to shift. We saw from wildfires, that's going to be different. But also disease risks are going to shift. The diseases that we have now are going to be different, and that, depending on where you are in the world, that risk might be a big significant aspect. And of course, again, this is from a hospital perspective. Building energy is a very big part of the carbon footprint of hospitals and the healthcare system. We also have to tension this with other air cleaning technologies, and every one of them has its pros and its cons. So HEPA filter devices are very easy, safe, etc., but they're noisy, they take up space, and they won't take very much out of the air in comparison to what GUV can do. We know upper room GUV is great, but it can be challenging to put in some spaces. We do have to think a little bit about chemistry on it. Ionizers, photocatalytic, there's a whole raft of these out there. I'll give you some plus points. They're quiet and they're often cheap, but do they inactivate microorganisms? I don't know. And if they do they do it by putting a shed load of chemistry into the air, which is a really bad idea? There's a lot of questions on those ones. And then where we are with 222, we know all the data shows so far it's safe in terms of skin and eye exposure if we do it right. The inactivation looks spectacularly good. I think we need to know a bit more there. It's quiet, but of course we have got that chemistry issue, and it's still an emerging technology, which has a bit of a cost barrier in some places. As we go further forward, maybe we can optimize things. And I just want to share this study from a few years ago, which was around upper room UV, where we were looking at, well, where do you put these and how much benefit can you get? And this was really quite conservative, because we said, okay, we've got a multi-zone hospital here, 6 zones. We've got an infected person in zone 1 who has TB. We'll look at the risk of infection, and we'll look at the energy costs in order to mitigate here. And we said, well, let's start. We've got 3 air changes an hour of ventilation, which is a little bit low from a hospital. So what happens if we double that to 6? 6 is the national standard in the UK. But what happens if we actually double it to 6 by putting upper room UV in instead? And actually, we know that you can easily go way more than that. This was a fairly conservative estimate. But what happens if you put it in just where the patient is? Or what happens if you put it in patient zones, or in circulation zones, or in everywhere in that one? And these are what we found. Sort of ignore the blue and red, because those are just how we did some of the zonal modelling parameters. But this is our baseline, 3 air changes an hour. And we're predicting numbers of new cases. I can't remember what period this was, but in this model. If you double the ventilation rate, you reduce your number of cases quite significantly, but you massively increase your ventilation energy costs. If you put your upper room UV in, stick it into just your risk patient zone, you're almost as good as improving the ventilation, but way less energy consumption in there. And then if we start sticking it in to some of the other spaces, we can actually get better than what we'd achieve by ventilation, but again at a lower energy cost. Now, we would need new data for this, we'd have to do this for 222. I think it'd be quite easy to do, look at different scenarios. But actually, these sorts of things allow us to start to look at those trade-offs and start to think about where those benefits might be. Last thing I want to say is, can we produce guidance yet? Where are we with this? I'm not sure we're at a point where we can produce an absolute standard on it, but I think we're in a position where we can say a heck of a lot more than we do now about 222. I think we obviously know what the TLV is, and from at the moment, the ICNA values seem to be a little bit safer in terms of particularly chemistry, but perhaps there's more to go there. We've established you can't just put in a completely unventilated space. We've established some of the practicalities around installation. I think we still need data to support how we could size things. And the graph you can see is from some of the modelling we've done based on the Leeds chamber data. And you can start to see that having maybe 4 lamps is almost as good as having 5 lamps. And you can easily get quite good inactivation within it. So we can optimize these, but we then need more data to get to different specific settings. So my final take home, Far-UVC has a huge amount of promise. I think we've seen that here, but there's still more data and evidence that we need to really make this robust. I think we can't see it as a magic bullet. It's got to be part of a layered intervention. It's got to think about all of the other factors in buildings and the air quality in those buildings. We've established all the key principles and they're robust. I think the science there is pretty good now, but we still have some gaps in some of the fundamental understanding, the real world, and there's bits we don't understand. And we've had some conversations about those. And then we need to start thinking through, how do we now make the case for this being cost effective, having those benefits, being part of that bigger picture of how we improve our indoor environments and be resilient for the next pandemic, as well as for our seasonal infections. And how do we use that to produce useful guidance that allows people to select this and use it correctly? And everybody here needs to be part of that. And I apologise if I've missed anybody's discipline here, because I was trying to think. I think I've missed electrical engineering out, sorry. And I'm sure somebody will come and tell me afterwards everything else. And if I was really clever, I'd have done a word cloud, but I'm not that clever, so I haven't. But we need everybody here. The brilliant thing about this conference is all the disciplines have been in the room. We've not just focused on a biological aspect or on a safety aspect. We've brought everybody together, even the bits which I'm sure every one of you has sat in a presentation and gone, that went over my head. Yeah, because we're all, all of us have had that bit where we've been outside of our comfort zone. But that's the beauty of what we've done here. And I'm going to say a thank you. So thank you to our team, people who we've worked with over the past few years, particularly Kenny, Ewan, David, Manuela, the other David, and all of our other colleagues back in Leeds and back in Scotland who we've worked on, people who've funded all of our research on this. And a big thank you to ICFAS community. You folks are brilliant, and that's what has made this conference really good. So that's me.
Well, I couldn't imagine a better wrap-up talk, so thank you, Kath. And we have time for just a couple of questions, but we're running a little bit behind. Right at the back.
Regarding the activation methods, how those differ to practice, that's something you're aware of.
I think so. I think David's lab's working on some of that, and I think it's certainly on many of our research priorities agenda. As with all these things, it depends on resource, time, and money. But yes, it is. I think it is an important priority, and I think there are people who are starting to work on that.
So one of the technical issues I don't think anybody's talked about here is there aren't any really good techniques for collecting viable viruses from clinical and general public arenas. The successful experiments are in the laboratory, but getting viable samples in the general public space is difficult. Can you comment?
Yeah, I agree. And I think that has got better over the pandemic, but I agree, in order to do it well, you need a very good sampler and you need the right expertise to do it. And it's not something that is easily done. And I'm sure there are others in the room who've experienced doing this who can comment on that as well, that it's a hard one to do.
Okay, thank you so much. And